My name is Alan Dietz. I'm the director of the Impact Lab, the Immune Progenitor and Cell Therapy Lab at Mayo Clinic and Mayo Clinic Rochester. The lab's purpose is to investigate the use of cells as drugs to the benefit of our patients, and especially for those diseases which are particularly difficult and refractory to other therapies. Other Mayo Clinic colleagues and myself, including Drs. Bill Fabian and Dr. Eric Dozwa, have had some success combining cells onto matrices for use for the treatment of anal fistulas and Crohn's disease. What we did was essentially take the patient's own mesenchymal stem cells and combine them and use dissolvable matrix, typically found or analogous to a dissolvable stitch, and used that, implanted that in the fistula of patients with Crohn's disease, and it had very good results. I think this was a groundbreaking trial in the fact that it demonstrated the effect of the combination of both cells and matrices to produce an unexpected and very profound response, positive response for the patients. We set about then to look for what other matrices could potentially be available for exploitation and combination of cells. The paper we are publishing in the Mayo Clinic Proceedings is Regenerative Materials for Surgical Reconstruction, Current Spectrum of Materials and a Proposed Method for Classification. As we reviewed the spectrum of materials that were available, it became clear that there is a large number of synthetic and biological materials currently under use and currently being used as part of clinical practice. These materials may have the capacity to be able to bind certain cell types, and actually the combination of living cells with these materials may actually provide additional benefits than the materials alone. The first step in that kind of effort is first to understand the types of materials that are available and a way to classify them. It's impractical to measure every cell material combination in a patient environment. What would make the most sense and most efficient use of resources would be to test classifications of materials, certain groups of materials with a certain cell type, and then using that to kind of either identify those areas where you want further investigation or eliminate those areas from further investigation. So we set out to get the entire spectrum of materials that are available, currently under clinical use, as with current FDA classifications as devices, put them into categories and groups, and then using those groups and categories, hopefully that will be a launch pad for individuals who are interested in doing cell matrix combinations for future therapies. We hope this classification will be of high benefit not only to clinicians as they think about using materials in certain indications, but also scientists to look at cell matrix combinations and to see if we can identify new therapies and new therapeutic potentials for these very important materials for our patients.
We hope you found this presentation from the content of Mayo Clinic Proceedings valuable. Our journal's mission is to promote the best interests of patients by advancing the knowledge and professionalism of the physician community. If you are interested in more information about us, our homepage is www.mayoclinicproceedings.org. There you will find access information for our social media content, such as additional videos on our YouTube channel or journal updates on Facebook. You can also follow us on Twitter. More information about healthcare at Mayo Clinic is available at www.mayoclinic.org. This video content is copyrighted by Mayo Foundation for Medical Education and Research.